In algae, we have studied the general characteristics, the methods of reproduction and their economic importance. And so far we have studied some of the life history of certain examples like nostoc, chlamydomonas, vaucheria, sargassum. Now we shall study about another example of red algae, rhodophyta. So in the previous aspect we have studied about the brown algae, that is Phaeophyceae class, example sargassum. Now in the Rhodophyceae or the Rhodophyta group or the division, we are studying example like Batrachospermum. Other examples are there like Polysiphonia and many others. Now Batrachospermum, this algae is a freshwater algae. Now this is found in clean water and unpolluted waters where there is somewhat a cool climate and where the water is not disturbed, in other words. So in such kind of fresh water, Batrachospermum red algae is found or occurs. Now the Batrachospermum algae, it is found usually attached to the rocks present in the water, either to rocks or any other hard substratum inside the water. Now in case of shallow waters, the algae appears somewhat violet green in color, but in case of deep waters, when it is found in deep water, it appears somewhat violet blue or somewhat dark green in color. So that is it is not due to the variation or the content of the pigment in the Batrachospermum, but it is due to the variation in the intensity of the light in the water. So that is how due to the variation in the intensity, the color differs when it is present in shallow water and in deep waters. Now this red algae, Batrachospermum, it is a multicellular algae. It is made up of many cells. So when we come to the structure of the thallus, and already basically we know that algae, the body of an algae or any alga, when you take it is a thallus. So even in bryophytes it is also thallus. So algae has not a well defined plant body, not having well defined root, stem or leaf, hence it is called as thallus. So it is a thalloid body. So in Batrachospermum the same thing occurs. So it is made up of a primary axis. This primary axis is made up of filaments. Suppose this is the primary axis, it is branched, and these branches are again unlimited. So there are this is the main axis which branches into the unlimited branches, lateral branches, and on each branch we find again short branches like this. So these branches are called as primary laterals. So the primary laterals are present on the main axis and also on the lateral axis branches. So this main axis consists of the elongated or cylindrical cells like this. So these cells are arranged end to end, and thus they form a row of filaments like this. So these row of filaments constitute the axis of that plant body or the algae, which gives rise to the primary laterals. So from the region there are primary laterals giving rise to, so these primary laterals are branched or unbranched. Sometimes they may be branched like this. So again, so such kind of the row of cells are present on all directions. So these lines what I indicate, it is nothing but the row of cells. These row of cells may be unbranched or they may be branched like this. So these cells are moniliform in appearance, but whereas the cells of the main axis consists of the cylindrical cells. And so this group of primary laterals or the cells of limited growth or the filaments of limited growth. So these filaments are of limited growth and they do not grow indefinitely, just like the main axis or the lateral branches, which are having also unlimited growth branches. So at this single region, these filaments arise and they form a globose structure. So this globose structure is actually called as glomerule. So glomerule is a group of filaments or the primary laterals they are. So they are having limited growth. These filaments consists of moniliform cells, but where these are cylindrical cells. That is what the thallus consists of. And when we study the structure of a single cell of this thallus, any cell when we take, but here the shape of the cells varies in case of primary laterals and in the case of main axis. And so this cell, it is a typically plant cell. That means it is having a cell wall, cell membrane, nucleus, cytoplasm and cell organelles. And the cell wall, as usual we have seen in the previous examples, the outer layer is made up of pectin, the inner layer is made up of cellulose. So that means the cell wall consists of pectin as well as cellulose. That is the composition of the cell wall. Then inside the cell there is already mentioned, that is the nucleus, cytoplasm and cell organelles. And more important, there is the Floridian starch, that is the reserve food material in the cytoplasm. Then in addition to that, there are pigment containing organelles which are called as the chromatophores. So the chromatophores are the organelles which are parietal in position to the cell wall, and they contain pigments like chlorophyll A and D. So there are different types of chlorophyll. So A and D is common in red algae, so whereas B and C is not common. So A and D, plus there is a blue pigment called as phycocyanin and also red pigment phycoerythrin. And it is because of this phycoerythrin, red pigment, the algae appears red in color. But however the content is more, and due to the presence of the chlorophyll, it is autotrophic in nature, means it is capable of synthesizing its own food material, and that is why it is autotrophic. That is, it is photosynthetic in function. So this is about the cell. So in the cell, the only characteristic feature is the presence of the Floridian starch or the granules which are present there. So these granules which are present freely in the cytoplasm, they are the reserve food material, plus the chromatophores contain chlorophyll, phycocyanin and this phycoerythrin, which gives the characteristic red color to the algae. Then that is about the cell structure. So the thallus is made up of the primary axis, and the base of the primary axis may have some cells which is attached to the substratum. It may be a rock or any other hard surface. So there is some holdfast like structure at the base by which it will attach to the substratum, and the primary axis originates, or it will arise from that part, and it will branch into many branches, and then each branch is having the short branches, that is the primary laterals, which are constituting a globose or a round structure called as the glomerule. That is about the this vegetative structure of the Batrachospermum. Then when we come to the reproduction of this algae, so there is only one type of reproduction, that is the sexual reproduction is very common in this. So sexual reproduction consists of the oogamous type of reproduction, and this reproduction is a somewhat advanced type of reproduction. So that means the organs which are produced, they are well developed when we compare to the other algae. So in other words, it is an advanced oogamous type of sexual reproduction. So the Batrachospermum may be monoecious or dioecious. So that means they may be born on the same thallus, or it may be born on the different thalli. So the male sex organ, that is called as the antheridia or spermatangia. So the male antheridia or spermatangia, also called as, so they are separately born, or they are born along with the female reproductive organ, that is called as the carpogonium, or sometimes it is called as a carpogonial branch. So the female is called as a carpogonial branch that constitutes the female reproductive part. So then coming to how these structures, how the antheridia are present, or spermatangia, which is also referred as. So we already have mentioned these two may be present on the same thallus or on a different thallus. That means it may be either monoecious or it may be dioecious in nature. Now this male and female reproductive organs, they are born on this primary laterals, the glomerule which contains the laterals there. So usually they are present on the tip of this filaments. So in case of the antheridia, if you take, suppose this is a particular filament. So this particular filament, it gives rise to antheridia or spermatangia. So each filament gives rise to about 3 to 5 spermatangia. So these spermatangia, they are born on the tip of the filament like this. So this is how the spermatangia are appearing on the filament. So each spermatangia is a single cell. It is a normal cell actually. Now this single spermatangia, it has a reserved food material. It is having a large nucleus, and each spermatangia forms a single male gamete inside. A single spermatangia produces a single male gamete. So now this single male gamete is released from the spermatangium. Spermatangia plural. So a branch may produce 3 to 5 spermatangia or antheridia. So each antheridium consists of single male gamete or this male gamete which is released. Suppose this is the male gamete which is released. As it is non-motile in nature, hence this is called as spermatium. So it is not having any flagella. So there are no flagella in this case of male spermatangia. So this is having a thick wall, and it is somewhat colorless in nature, and it is somewhat spherical in nature. So it is liberated from this spermatangia. So the spermatangia has a thick wall. So the wall will rupture and liberate this spermatium, which is so called, and it is never called as a sperm because of the lack of the flagella. So that is about the structure of the antheridium. Now next, after the sperms, this spermatia are formed, which are released freely. So these are not having any power of locomotion. So they move according to their own movement usually by the water currents, and they reach the female carpogonial branch. Here in the female carpogonial branch, what happens? Suppose these are the filaments. This is the filament. Now in the female carpogonial branch, the female reproductive structure is produced at the tip of this filament again. Now in this group of 4 or 5 cells, so where it divides, the terminal cell usually functions as the female gamete. So that forms the carpogonium. So this forms the carpogonium, and this carpogonium, it is having a particular structure like this. So it is a different cell. So there are 4 or 5 cells in the carpogonial branch. Actually, initially, when we say that the female reproductive organ, it is the carpogonial branch, it is having about 4 or 5 cells, and out of this the terminal cell will form the carpogonium, and this carpogonium is the female structure. So this is the carpogonium. So this total filament is called as a carpogonial branch. So carpogonium is actually the female reproductive structure, and it is actually the terminal cell which is referred as carpogonium. Now in this carpogonium, the nucleus divides into 2. So the one forms this egg cell, and actually there is single nucleus, egg nucleus. So this basal part, it is the egg or the oogonium. So this terminal part, which is having colorless, it is called as a trichogyne. So the oogonium, this is actually the oogonium, which consists of the dense cytoplasm and the nucleus, that is the female nucleus. The narrow part of the carpogonium, the terminal part, which is somewhat extended, it is called as the trichogyne, which is having colorless cytoplasm and no nucleus. So that is about the female reproductive part of the batrachospermum. So this batrachospermum produces the male and female reproductive parts in the glomerule or in the primary laterals, the filaments of limited growth. Then the fertilization, how it takes place. So the spermatia or the spermatia which are released from the spermatangia, or the antheridia, they are released, and when they come in contact with the trichogyne in the water currents, now this trichogyne is having a somewhat mucilage material on its surface. Now because of the mucilage material, the spermatia will become attached to the trichogyne. So when the spermatia or the spermatium, when it is carried in the water and comes in contact with the trichogyne, so it will stick up to the trichogyne because of the sticky nature due to the presence of the mucilage material on the surface. And this spermatium and the trichogyne, when they come in contact, so the wall of contact where the wall of the spermatium and the wall of the trichogyne are in contact, so that part gets dissolved. It dissolves so that the male nucleus enters into the trichogyne. So when it enters into the trichogyne, so this male nucleus, actually it divides into two parts. So that male cell or the nucleus of the spermatia, so it divides, and it divides into two, so that one of the male nucleus fuses with the female nucleus. So the remaining nucleus remains, it gets remained in the trichogyne region. So one of that, it will fuse with the egg nucleus, and it will fertilize to form the zygote. Now after the formation of this zygote, so here a mucilage is formed and blocks this region. So it is called as the mucilage block, or the plug it is called as. So it separates the trichogyne as well as the zygote nucleus, or where the egg nucleus is present. And then the fertilization takes place in this region. So the zygote nucleus is formed here. So when the zygote nucleus is formed in this region, this nucleus, it will divide by meiosis. So meiosis takes place in the zygote nucleus. So suppose this is the zygote nucleus, and the trichogyne gets separated, so the zygote nucleus remains. So this zygote nucleus remains here, and it divides into four cells by meiosis. Suppose these are the 4 cells which are formed from the zygote. So there is meiosis taking place in the zygote after fertilization. So this is the zygote which is formed. So the spermatia or spermatium, which contacts the wall of the trichogyne, so that wall of contact gets dissolved, so that the male nucleus will enter into the trichogyne, and that male nucleus is carried downwards, or it moves downwards, where it will fertilize the zygote. So the spermatia, the male nucleus enters and moves down. So it divides into 2 daughter nuclei, which I have already mentioned, and one of that will fuses, and the remaining remains in the trichogyne. So the male nucleus divides by mitosis, actually, not meiosis. So out of the 2 nuclei, one will remain in the trichogyne, the other will fuse with the female nucleus to form the zygote. And this zygote, next it will divide by meiosis, resulting in 4 nuclei. Now these 4 nuclei further divide by mitosis, and these nuclei will form a filament like this. They go on dividing to form filaments. So these filaments are called as Gonioblast filaments. They are called as Gonioblast cells. So Gonioblast initials, these are referred as the Gonioblast initials. The 4 cells which are formed from the zygote, they are resulting from the meiosis. So they also further divide, and when they further divide, they form a row of cells, a filament of cells, and they are called as the Gonioblast filaments. So the Gonioblast filaments, they further go on dividing, and then the terminal cell of the Gonioblast cells, these filaments, they give rise to a structure which is called as the Carbo Spore or the Gonioblast filaments. So that is the terminal cell of each filament is called as the Carbo Sporangium. Suppose here, if you take, for example, this is the Gonioblast filament. So the terminal cell of each filament, it forms a cell which is called as Carbo Sporangium. So this is Carbo Sporangium, but not Carpogonium. So Carpogonium means this structure. So this complete structure we refer as the carpogonial branch. See, the carpogonial branch gives rise to the carpogonium. So the carpogonium means it is a female reproductive organ. And after fertilization, the zygote divides by meiosis and gives rise to the Gonioblast filaments. So the Gonioblast filaments, which are formed from the Gonioblast initials. So the Gonioblast initials means the cells of the zygote which are resulting from meiosis, and then in each Gonioblast filament we come across the Carbo Sporangium. So this Carbo Sporangium gives rise to what is called as the Carbo Spore. So as a single spermatangium gives rise to single male gamete or the spermatium which is non-motile, same way the Carbo Sporangium gives rise to a structure which is called as Carbo Spore. So the Carbo Spore is a unicellular structure, and this Carbo Spore is released from the Carbo Sporangium. So the Carbo Spore, when it is released, next what happens? So there are further stages of this. The Carbo Spore germinates and gives rise to a tubular structure to form a filamentous structure. So the germination of the Carbo Spore takes place in suitable conditions. So the Carbo Spore or the Carbo Sporangia which is formed here, this is the Carbo Sporangia. Now certain cells of the Gonioblast filament, this filament, it also produces some non-fertile filaments, but not Gonioblast filaments. So these filaments, they protect the Gonioblast filaments, and the Gonioblast filaments and the sterile filaments surrounding the Gonioblast filaments, it constitutes a structure which is called as carposporophyte. So this is completely called as carposporophyte, which includes the Gonioblast filament and the sterile hyphae, or sometimes this is also called as the cystocarp. So cystocarp is the structure which contains these filaments which are resulting from the meiotic division of the zygote. And each filament, the terminal cell of each filament forms the Carbo Sporangium. As here the terminal cell of this forms the Carpogonium, here Carbo Sporangium is produced at each filament. Now one Carbo Sporangium produces one Carbo Spore. So that Carbo Spore is released. So the Carbo Spores are released from each Carbo Sporangium. So the Carbo Spores, when they are released, they are also non-motile. They are having a well protected wall there. Suppose when they come to rest on a particular substratum, they start germinating into a branched filamentous structure. So this branched filamentous structure will go on branching like this. So these are actually the filaments cells which are produced from the Carbo Spore. So the Carbo Spore will germinate, and again it will form a filament that branches and also further branches many, and this appears like a protonema, or somewhat it is heterotrichous in structure, and then it will produce a branched structure. Now this branch structure, which is made up of all filaments, so this is called as Chantransia Stage. This is referred as the Chantransia Stage. So this stage is called as Chantransia Stage because now when the Carbo Spore germinates into a tube-like structure, so that goes on producing many cells, and they form branched filaments. So this branched filamentous structure, which is somewhat having a creeping appearance, it resembles an algae which is called as a Chantransia. For example, in the previous one we have studied about Gonioblast stage, Palmella stage, and like that. So Palmella stage means it resembles Palmella algae. Gonioblast it resembles Gonioblast. Similarly, in Batrachospermum, we come across such kind of stage where the carpospores which are present. So the carpospore which is released from the Corpus Porangium, after having rest on a particular substratum, it germinates to give rise to the Chantransia stage. Now from this Chantransia stage, we come actually the asexual reproductive part of the this algae. What happens next? Now this Chantransia stage, when it expresses the asexual reproduction, it produces what are called as the monospores. Monospores are produced by the Chantransia stage. There are structures which are called as the Monosporangium. So this Monosporangia, again they are the terminal cells of the filament. Now suppose if you take the filament like this, the terminal cell of the filament, it slightly swells like this, it bulges, or in other words it enlarges. After the enlargement of the cell, this is called as a Monosporangium. So this Monosporangium, this is the Monosporangia or Monosporangium single. So Monosporangium is the terminal cell. See here, whenever we see this reproduction stages, almost the terminal cells, they involve in the reproduction part. Even in the case of this male, the terminal cells give rise to the spermatangia, and even in the carpogonium, the terminal cell of the carpogonial branch gives rise to the carpogonium, and even in the Gonioblast filaments, which are resulting from the zygote, meiotic division of the zygote, there also the terminal cell gives rise to the Corpus Porangium and also produces the carpospore. The carpospore gives rise to the Chantransia stage. The Chantransia stage, which is having again many filaments. So the certain filaments, now especially the filaments which are having short lateral branches. So not all branches of the Chantransia stage gives rise to the monospores. The short lateral branches of this stage, they involve in the production of the Monosporangia. So the Monosporangium is formed at the tip. The terminal cell will become swollen, so that terminal cell of the short lateral branch, it will become swollen, and each Monosporangium gives rise to a single monospore. Now here also each carpogonium gives rise to single carpospore. Similarly, each Monosporangium in the Chantransia stage gives rise to a single monospore. So the single monospore is developed from the Monosporangium, and that monospore, it also germinates to give rise to another Chantransia stage. So like that, Chantransia stage is the repeated. So it is not only once formed in its life cycle. So it is formed many times, and after certain period, at one stage, the lateral branches of the Chantransia stage, they filaments which are erect in appearance, some of the filaments which are erect, so they give out lateral outgrowths and gives produces the Batrachospermum plant. That is how the asexual reproduction takes place in the Chantransia stage, which does not involve the sex organs there, and it involves only the spores which are produced in this case. So that is about the asexual reproduction. So the sexual reproduction is somewhat advanced when we see the structure and all that, because the organs which are produced, they are well developed when we compare to the previous examples like Wacheria and even in this Chlamydomonas and all that. So there the example, this is the well developed. So the red algae, the rhodophyta, are developed when we compare to the green algae and the brown algae. So even in Sargassum also, we don't come across such kind of structure like this. So this antheridia which are produced, and either already we had mentioned the Batrachospermum is either monoecious or dioecious, where the two organs are present on the same plant or on the separate thallus, separate thalli. So the spermatangia produces spermatium, which are carried, as they are non-motile, so they are carried passively by the water currents, and when they come in contact with the trichogyne of the carpogonium, they become attached. So this is because of the mucilage layer present on the surface of the trichogyne, and then the fertilization takes place in this region. The male nucleus divides into two, one of it fuses to form the zygote. The zygote divides meiotically to form 4 gonioblast initials, they are called as. Now the gonioblast initials produce the filaments called as the gonioblast filaments. Now in each filament, the terminal cell becomes differentiated into Carbo Sporangium. So it is due to the accumulation of the reserve food material and dense cytoplasm. So there the Carbo Sporangium produces a single Carbo Spore. So these Carbo Spores which are produced in the Carbo Sporangia, they are released, and when they come in contact with a hard surface like rocks or any other substratum, they rest on that region and give rise to a elongated tube-like branched structure, filamentous structure, which further creeps over that surface, and that is called as the Chantransia stage, which resembles the Chantransia algae. The Chantransia contains, or also again the filaments of the Chantransia, especially the short lateral filaments, they give rise to the Monosporangium. The terminal cell becomes swollen, enlarges, and becomes the Monosporangium. Each Monosporangium gives rise to single monospore, and that monospore which is released, it further gives rise to another Chantransia stage. So like that, in a Chantransia stage, the short erect filaments of the Chantransia stage, they further develop into lateral outgrowths and gives rise to the Batrachospermum red algae. So like that, the Batrachospermum is found only in clean water and cool climate water, and even fresh water there. So that is how the Batrachospermum reproduces. And the cell structure already I mentioned, the unique feature of this cell is only the presence of the Floridian starch and the chromatophores which contain the chlorophyll, phycoerythrin and phycocyanin. They are the pigments which are present. Due to the chlorophyll, it is photosynthetic, and due to the presence of the phycoerythrin, so it appears this red in color. So hence it belongs to Rhodophyta or the green algae. So that is the life history of Batrachospermum.